फार स्लो केलंय का?
Uh, Hello, Ashwini, am I audible?
सोनल
Yes, Sonal, you're audible.
Yeah.
Okay, sir.
Yeah.
अच्छा जय he has joined?
Yes, sir, he's there.
Sir has
Yes,
joined.
I'm right here. I came in 10 minutes
Sir,
early
sir,
on.
namaskar.
Namaskar. How are you guys doing?
Namaskar. How are you, sir?
Doing good. Doing good. Yeah.
Kurkure
So,
sir has also joined.
you are going to visit India?
Yeah, I was planning to come in the month of May, Ravindra, but unfortunately the cases were so pretty high in Mumbai. I postponed my visit. I'll try and come in August. Definitely in August I'll be there. Yeah. Okay. Yeah. Hello, Dr.
Audible, sir.
Kurkure. Yeah.
Director of Research, Kurkure, sir.
Namaskar. How are you
Namaskar.
doing? Probably you're
Sir,
muted.
unmute yourself. Kurkure, sir.
Thank you. Sir, we
Sir,
were
yeah,
also facing the same thing, but 2 months before we were not sure how the things will be. But now it's good that with the grace of God and few efforts, it has been controlled now. And by the time you will come, probably that will be the least incidence of this pandemic.
Yes, look forward to that. Look forward to that. Yeah.
Great, doctor. सर आपलं नायबचं अजून कास्ट चालू झालं नाही सर. सर अनम्यूट करा.
या आज दुपारी होते ना प्रेझेंटेशन.
हो सर ते पण अजून ते IDPच चाललंय.
ओ माय गॉड. मग दोघेही कोइन्साइड होतील का काय?
काय समजणं झालंय सर. ते चालू ठेवलं मी.
तुम्ही हे सुरू करून द्या मग आम्ही इथे काही प्रश्न नाही मी पाहतो तुम्ही तिकडे कारण ती पण बॅटिंग करणं जास्त महत्वाचं आहे.
येस सर.
शक्यता
इथे डोंट वरी नाऊ आय एम हिअर डोंट वरी.
ओके.
आणि इंगोले सरही आहेत ना पण मला वाटतं सगळ्यांना तिकडे जॉईन व्हायचं आहे. ऑल
येस सर. ऑल PI को PI they have to
ओके
join.
डोंट वरी आय एम हिअर यू डोंट वरी.
ओके सर ओके. Hello. Sir, namaskar. Sir, how about COVID here right now situation in US?
It's much better. We don't really have to wear masks when we go out anymore.
Very nice.
I think we are, especially the university town which we are here, I think we are close to about 70% of the population is vaccinated.
Oh.
A very high rate of vaccination going around in the US. So hopefully we should another 2 months time gain some level, not sure how much, but some level of herd immunity coming into the process.
Okay. Actually, what happens, sir? I think the participant, they knows that in the morning only the seminar is, but that is why the people, they are taking more time to join.
Yeah, no worries. As long as do you want me to text Suresh and say we are running a little late, or
Okay.
do you want me to send him a message?
No worries, sir.
No
I
worries.
think it will
Okay.
be 10 minutes late.
Okay.
Yeah,
That's fine. Yeah.
And sir, I have one parallel meeting is going on. The same
Okay.
project, NIHEP, that is a technical review meeting.
Okay.
So actually, it is
Yeah.
about to start at 2 o'clock, but it is a delaying because the other project they are presenting and they are evaluating
got
the experts. So I will just start and I will be there. Dr. Kurkure sir is here
Yes.
as a chairman for the session.
Yeah, I did overhear your conversation about another parallel meeting. Yes.
Yeah.
Sir, namaste, sir.
Namaste, how are you?
Yeah, I'm fine. Thank you, sir. How are you, sir?
Doing good. Good to see you again. Yeah.
Thank you so much, sir. Yeah. Nice to see you.
Hello. Right.
Yes, sir.
Dr. Vilas and Dr. Invoicing, again, you have to just on the message in box, that please join all the participants immediately.
Yes, sir.
Yes, sir, I should. Right. They are very punctual. It's not group this.
Vilas, Ravindra, are you recording this?
Yes, sir. Yeah.
Okay.
Yeah.
And that will be available for
Also,
others.
webinars, all the seminars are international training programs. What we have done, they all are recorded and we have put up on our website. We have
Okay,
separately
excellent.
formed for the NHB. Yeah.
Excellent. Yep.
So any candidate, any students, faculty, they can see again and again.
Right.
So they can go to the website and see the videos.
And how is your BSL-3 coming along?
It will be ready because some of the work is going on. Some equipments they are setting up. And
Right.
I have also received, sir, ICP-MSI and LCMS-MS also. So the installation work is going on. But because of this lockdown, some people they are finding difficulties. Some engineers for putting that gas line, the cylinders,
Right.
and the engineers. And these are the multinational companies, sir. So they are not allowing their people to go out. From home, but I am requesting them to complete within a month so that we can start the research work also.
Right.
98% that all procurement work we have completed so far.
Good. Very good.
That is what I'm just reviewing.
Okay.
ICR authorities and World Bank people, they are also there for assessment of this
Right.
project.
Okay.
Yeah.
So they have some set of metrics on which they evaluate the program?
Yes, yes. They have their own standard protocol and
Okay.
format they have given, and with that format, they are just discussing all the objectives with the target and achievements and what is the outcome. So everything.
And will they come back with, will they come back to you with additional recommendations and changes? Do they
Yes,
typically do that?
Yeah, yeah. They look to that. So there are some 3, 4, 5 ex-vice chancellors are there. They are expert committee members. So they evaluate each and critically.
Oh, okay.
Yeah.
Good.
It's 123. The 400 participants they have registered. And
I saw that
we
in your
have
email.
about,
I was very surprised.
Yeah.
Very surprised.
yes. And
So
the
do
students
you,
and even so not only from our MAPSO, they across the
Okay, from
country,
other
all the
places.
veterinary universities. Yeah, yeah.
Very good.
And the faculties are also more. Teachers.
Excellent.
Dr. Jende, I think we can start because it is already 8-10 minutes. Time has exceeded.
Yes, sorry.
We don't want our
One-
learned speaker to wait.
Sir, another 1 minute.
Okay, fine, but we will start.
Okay. Dr. Sonal. Dr. Sonal, you can start.
Yes, sir. Okay, sir.
Yeah.
A very good evening to all the participants, colleagues, the chairman, co-chairman, and the team members at Naheb Cast Mafsu at Mumbai. And a very good morning to our speakers for today, Dr. Bhushan Jairao sir and Dr. Suresh Kuchipudi sir, who have taken efforts to discuss with us or to spend time with us today. I welcome you all for this third session, or I should say the third day of our training program on gene cloning on advances and applications in veterinary sciences. Without wasting much time, I'll introduce you all to the speakers for today. So friends, today we have Dr. Bhushan Jairao sir, who is currently working as a resident director of at Penn State Animal Diagnostic Laboratory, and he's also the professor of veterinary and biomedical sciences. Sir has joined Penn State in 1998, and since then he has been actively involved in developing the diagnostics, extension and research programs in order to take care of the antimicrobial resistance, animal health, emerging and infectious diseases, and also food safety. Being the subject matter expert for the government of United States, he has advised public health agencies on issues related to capacity building and establishing surveillance programs for emerging zoonotic diseases. He's also the coordinator of the One Health Minor Animal Diagnostic Laboratory at Willie Lane University. Sir is also very actively involved in the administrative roles with the United States Department of Agriculture and American Veterinary Medical Association. His research group basically focuses on epidemiology and pathogenomics of animal, food, and human pathogens. Sir has till now mentored 21 PhD students, 14 master's students, and 11 postdoctoral fellows, which also includes some students from institutes of India. Sir has reviewed and authored, Sir has authored around 120 papers in peer-reviewed journals. And he also serves as a consultant advisor to many colleges of veterinary medicine, diagnostic laboratories, and in Africa, Eastern Europe, and Southeast Asia. Today, friends, sir is going to talk about the detection of food pathogens and animal disease diagnosis employing recombinant DNA technology. Now, for this particular session, the chairman for today will be Dr. Nitin Kurkure, sir, Director of Research at Maharashtra Animal and Fishery Sciences University, Nagpur. And he will be accompanied by the co-chairman, Dr. V.V. Deshmukh, sir, Professor, Department of Veterinary Public Health, College of Veterinary and Animal Sciences at Parbhani. So I now request the chairman and co-chairman to please take over the session.
Thanks, Dr. Sonal. Thanks, Dr. Sonal. I'm Dr. Kurkure, chairman for this session today. And along with me, Dr. V.V. Deshmukh, who is Professor of Veterinary Public Health at College of Veterinary Science, Parbhani, along with me. Dr. Sonal has introduced about the first speaker today. It's a very brief introduction, and I'm very sure that it is not possible to give a bird overview also in one slide about the speaker today. Anyway, it was there are a lot many things which can be included into it, and that will be helpful for others also to have the future links with him, collaborations with him. And today, in fact, I am also very much lucky, and I thank the organizers for giving me a chance to be a chairman of the session in which Dr. Bhushan Jairao and Dr. Suresh will be speaking. So it's an honor to me to be chairman of the session in which these two speakers are there. So without taking much time, I think Dr. V.V. Deshmukh will also agree with me for this. Without taking much time, I don't want the learned speaker to wait in these formal things, and we will be more benefited by listening to him. And so I request Dr. Bhushan Jairao sir to please start your presentation. Time, Dr. Jende has already communicated about the time, but time is with you. As you feel, you can take that time. I would request Dr. Suresh, if you need some more time, I would request him to wait for some time, and then we can start his lecture. So over to Bhushan Jairao sir, please.
Okay. Thank you, Dr. Sonal, for that very nice and kind introduction. Always there to be come back to Bombay Veterinary College and MAFSU. It runs in me for the last 42 years, and I cannot separate myself from the BBC. Oh, now it is MVC, by the way. So I have to get used to that. So very, very much in the game process here, and also would like to take the opportunity to thank Dr. Kurkure, Dr. Deshmukh, Dr. Ravindra Zende, and the organizers for inviting me, and of course, my colleague, Dr. Suresh, and he will come and join you guys later on to share with you some of the things that we have been doing here at Penn State as it relates to recombinant DNA technologies in our DNA-based technologies. So the focus of my talk today, as Dr. Zende has asked me to do, is to talk about the detection of foodborne pathogens and diagnosis of animal diseases using rDNA technology. Now, the slide that I have in front of you here is a picture of our Penn State Animal Diagnostic Lab. And we are close to about 60 members in the diagnostic lab. We are close to 12 board-certified veterinarians and the remaining technicians. We process anywhere between 16 to 19,000 accessions each year. That relates to about close to around 200,000 samples that flows through our lab. We do necropsies of close to 700 large animals and about 8,000 birds on an annual basis. So that's what we do. Our primary focus is to provide diagnostic services to the Commonwealth of Pennsylvania. Now, we are not the only lab in the state of Pennsylvania. We are a part of a bigger picture in the Commonwealth that we are associated or a part of the Pennsylvania Animal Diagnostic Laboratory System. So if you look at the Pennsylvania Animal Lab System, there are 3 different labs that are a part of it. One is the Pennsylvania Veterinary Laboratory Systems, which is in Harrisburg. The next lab is at the University of Pennsylvania, or UPenn, close to Philadelphia. And the third one is our diagnostic lab. That's the Animal Diagnostic Lab located close to State College. As mentioned earlier, our primary function is to provide diagnostic services, but we are also very actively involved in doing surveillance programs at the federal level and at the state level for largely to monitor some of the infectious diseases of food animals. We also spend quite a bit of our time developing new diagnostic tests, training and educating new diagnosticians, veterinarians, and graduate students. And of course, we are very much into doing translational and applied research. So with that, let's move on and focus on what we will need to be talking today. So the way I have set up my talk today is in the form of a 5-act play. I'm sure you must be all lovers of going to the theater. So the title of my play is called as The Queen's Gambit. Any chess players here? Okay, so the title of my talk is, which I'll be sharing you, is based on a theme called as The Queen's Gambit. And the first act is to know thy enemy. Who is your enemy? And the way we will approach it is using a case study where we'll talk about Dr. Sharma's RT-PCR assay. Then we'll dive down and look into the concept of a logic model that I use, is addressing issues such as who, where, why, when, how, and why. And then, of course, we will distill everything down into the analytic continuum. The second act is who should lead. Should lead by the pawn or the horse, especially if you're doing a Sicilian defense right in your first move. What is E5 or C4? So that's what we're really talking about, is what are some of the bacterial identification systems that were developed, and what were some of the typing systems that emerged all side by side with the identification systems. The third act is think before you move. Is phenotypes, genotypes, or what would you like to do? Like to do both? Or where's your focus? The fourth act is, we have called it as Antaryami. That means genomes and genes tell it all. And the fifth act is checkmate. I know who, what, and where you are, when and how you turned into our enemy. Hi, Suresh. So this clearly shows I've laid out a game plan that I would like to walk you through before we come and answer the basic question. So what is it that we are really looking at in terms of developing an ultimate diagnostic tool?
Good morning.
Sorry. So the first act is know thy enemy, and let me start off with the first act. And generally in a theater setting, the first act is introducing the players, laying out the big picture. Where are we taking you in terms of the theater or the acting? Okay. So let's start off with a case study here. There's a national study that warns the possibility of a new pan-resistant enterotoxigenic E. coli O11H7 from chickens. And who is the reporter? It is nothing else but Bhushan himself. And what does this newspaper write-up tell us? It says a new emerging pan-resistant enterotoxigenic E. coli O11H7 from chickens has been identified in China, Vietnam, Thailand, Malaysia, and India, of course, Maharashtra. Although experts say that there is no imminent threat, this strain has become more infectious to humans and needs to be watched closely in case it becomes a potential pandemic bacteria. So there are other findings which also suggest that poultry workers in China, Malaysia, and India have shown excessive rates of gastroenteritis and elevated morbidity rates. Close monitoring of human populations, especially workers in poultry industry, should be urgently implemented. Now that I've set the stage for you, let's see what happens next. So let's set the scene here. The Ministry of Agriculture asks Dr. Sharma, a very famous veterinary microbiologist and immunologist, to provide them with a highly sensitive and specific diagnostic test to screen poultry, farm workers, chicken meat, and animal waste for E. coli serotype 011H7. So, Dr. Sharma, what is your strategy and tactics to get this done? Do you have a game plan? Okay. Let's try and see what Dr. Sharma does in the next set of slides. So the first question will be ringing in his mind is, do I have the correct stuff to meet the mission of what I've been asked to do by the Ministry of Agriculture? So that's really, he's fighting an inner struggle to find the right way. So what does he do? Behind the curtains, Dr. Sharma assembles a team of researchers and assigns them to develop a diagnostic test. Just like most of us who administrators, we have become administrators, we go back to the lab and say, hey folks, develop a test, okay? So he gives them 7 days to develop an RT-PCR assay, and at the end of the 6th day, his team develops an in-house E. coli 011H7 diagnostic assay. So Dr. Sharma is really happy. He takes them out for lunch. He calls up the ministry and tells them, hey, by the way, I've developed the test. So the ministry is very happy. And of course, now the test is being offered, and Dr. Sharma is awarded a medal by Dr. Paturkar, the Vice Chancellor of the university. 1 month later, OMG, something is not right. In fact, many things are not right. Problems and problems and problems. So what were the problems? These were some of the major issues that were identified with the test that was being offered. One is the sample that is delivered in person tend to be of better quality than that sent by mail. Why? Why is it? Why do fecal sample bags leak? Why is it happening? More importantly, the PCR controls and standards vary in the CT cycles. And is this okay? Negative controls at times are positive, and what should we do about it? We have a high volume of call from clients on interpreting PCR results. Why? Of course, all of these things are really hitting at very much at the base of the assay. So what happens? The Vice Chancellor calls Dr. Sharma and says, hey, better suspend your test till we get this right. Guess it's Miller time. In the US, when we say Miller time, that means it's time for a beer, okay? It's too early for me here. Probably it's the right time for you there. So with that, let's ask the very primal question. So what went wrong with Dr. Sharma's PCR test? I'm sure all of you have generated some fantastic ideas and thoughts, but let's try and answer this with the help of a Chinese philosopher he was there for almost 3,000 years ago, Sun Tzu, who stated very clearly that strategy without tactics is the slowest route to victory. And tactics without strategy is a noise before defeat. So let's try and use the concept of strategy and tactics to understand what really went wrong with Dr. Sharma's PCR test. So before we do that, let's try and understand a logic model that I would like to share with you. Being trained in epidemiology, I generally go back to my basic questions: who, where, what, when, how, and why. Okay. In order to answer that, I have to ask these specific questions. Who are our enemies? Now you assume yourself to be a chess player. You're looking on the opposite side of the chessboard. You're saying, who are my enemies? Is it bacteria, viruses, parasite, toxins, and protozoans? All of them or some of them? Okay. Where does our enemy come from and where does it go? Does the enemy utilize multiple vehicles and transmission pathways? The next question is what? What does the enemy do to us? They target the weakness in our system. When does the enemy like to attack us? The question is very simple, but we know that when we let our guard down, we are more vulnerable to attacks. The last question is how and why does our enemy attack us? We give them the opportunity to do so. That's what we really know about it. So how do we translate the query, the question, and the knowledge that we have at this time in order to develop our strategy and tactics? So what is the strategy and tactics when we want to identify, when we want to know who are our enemies? Is the first thing is to do is to accurately identify them. Next is when we want to know where does our enemy come and where does it go, we need to subtype them, especially understanding their serotype, the genotype, and then differentiating into the process of saying whether they're the good, the bad, and the ugly. Next is what does our enemy do to us? We need to understand what are some of the virulence factors our enemies have with them, or weapons, in other words. The last one is how and why does our enemy attack us? So in order to prevent an attack, our best option is to develop a monitoring or a surveillance system, okay? So if you have the ultimate all-in-one diagnostic tool, that will allow you to not only identify the species, the serotype, the genotype, the antimicrobial resistance genes, the virulence factors, and mobile elements. That will be the awesome thing is you take a sample, you extract whatever you need to extract in terms of nucleic acid, and if you're able to generate a whole wealth of information, you're all set to go. So the question here I'd like to leave up here is, do we have such a diagnostic tool? With that, let's move on and try and fully understand what do we mean by an analytic continuum. Generally, most of us, I'm sure 99% of us truly get excited when we develop a diagnostic test, okay? It's largely how well can I detect, what is the CT values, what is the threshold of dissection, how sensitive, how specific it is, how does it work in a set of given controls, within positive controls, negative controls. We really get excited and spend an ad nauseum amount of time trying to work out the analytics. In fact, I have stepped back and I personally think that's a very boring job because it is nothing else but it's taking a cookbook and trying to make something out here. But what really makes a analytic continuum is we tend to overlook one of the parts of the continuum, what we call as the pre-analytic part of it, and the other one is the post-analytic. We spend a lot of time doing analytics, but we do a very poor job in addressing the pre-analytic and the post-analytic issues. So let's dig a little deeper and say, what do we mean by pre-analytic? When it comes to pre-analytic is how do we collect the sample? How do we transport the sample? And once the samples are received, how are they processed or accessioned into the system? That we call as pre-analytic. The analytic component comprises of processing of the sample, especially when it comes to a nucleic acid-based system. Is the next step involves DNA and RNA extractions, doing PCR assays, or going and doing a whole genome or next generation sequencing. So I'm sure most of you folks have heard the word garbage in, garbage out, right? If you have a very poor sample that is not collected well, that is sitting out for a long time, not properly refrigerated, it is thrown in the mail, comes in to the lab, it's totally lost. We don't know anything to do with the sample. So the concept of garbage in, garbage out becomes very relevant in a diagnostic system if we don't give adequate attention as to how we collect, transport, and receive the samples in the lab. That's the first and the foremost we need to do, is to assure the quality of the sample that's coming in. The next is the analytic part of it. And finally, the post-analytic is how do you capture the data, the analytic data that is coming? How do you review Levy Jennings charts? How do you incorporate the data that you've generated into a laboratory information system? And then finally, how do you report it to your clientele? Now here, remember, your clientele, all of them may not have a PhD or a DVM. They may be clientele who may have a 4th or a 5th grade level education. How do you convey the meaning to them? How do you convey the findings of the result? Okay, so those are some of the things that we really need to look into when we develop a report for a clientele. So let me now switch over and talk about how do we apply this analytic continuum in a diagnostic setting. So here I'm sharing with you a decision tree for the bacteriology section of the Animal Diagnostic Lab. So from where do we get our samples? We largely get our samples from our pathologists who are involved in doing necropsies. We receive a lot of pathologic tissue samples, aborted tissues, whereas we also get a lot of samples from our external clients, veterinarians, producers, researchers, and can be a whole plethora of samples, ranging from milk samples, fecal samples, swabs, all the way to environmental samples. And more recently, we do get quite a few fish samples in our diagnostic system. As soon as the samples are received and they're accessioned and logged in a case report, a number is being created, then each of the sections are then notified. The first step is to look into what is the request that is being made. Are they looking for a specific pathogen? Are they looking for, say, for example, salmonella in feces? Or there is no instructions, you just would like to see what is there. So if there is a specific request for a pathogen, we typically utilize a blood agar media with specialized media. Whereas if there is no request, usually it's a blood agar plus any other media that the bacteriologist section feels is relevant at that point in time. Following which, we identify the bacteria using MALDI-TOF. We then look at the antimicrobial sensitivity using antimicrobial resistance system, that is the phenotype-based system. And then we, when requested, we also look for virulence factors using RT-PCR-based assays. So when we talk about pre-analytic, comprises of two very important areas we need to address. The first one is the samples. The majority of the samples that we get in a diagnostic lab, I would say up to 65 to 70% of the samples, they come through mail. Whereas the remaining 30% of the samples get delivered in person by the clientele or when they bring in their animals for necropsy. So as soon as the samples come into the lab, we log in the samples, we assign a case number, we assign a case coordinator, and then we notify the sections that to let them know that they will be expecting samples to come in for analysis. When we talk about the analytic phase of it, how do these samples go through? What is the workflow in the analytic part of the continuum? The first and the foremost, any diagnostic lab that is giving clientele-based services must have the following three standard operating protocols with them. One is, how do you do DNA and RNA decontamination? Do you have the protocols? How do you process your samples? We would prefer to go using a magnetic particle processor because it reduces cross-contamination, it is fast, it is accurate, and less errors that that get induced when you're doing physical extraction processes. And the last and the foremost is, we always rely upon a certified or a trained technician who has undergone all of the proficiency testing to perform the test. We just can't assign a graduate student or a technician who doesn't know much about PCR to do an analytic test, okay? So keep this in mind. In order to have a very successful PCR laboratory or an analytic part of the laboratory, you need to have your tactics. Your tactics is decontamination, how do you process, and who does it. Having laid the groundwork for the analytic portion, let's now talk about how does the sample typically move. The sample comes into the first area, area 1, which we have called it as the dirty area, where it is received, accessioned, and then processed. It then moves into the DNA and RNA, from the DNA and RNA extraction room into the clean room. So when it's in the clean room, we bring in another component of the analysis, is the master mix, that is created in a separate lab that is away from area 1, 2, and 3. So the reason why we do this is to prevent cross-contamination and prevent contamination of reagents, okay? So once we bring in the master mix into the second area of clean room, they are mixed and then taken over into the clean area for doing PCR-based assays. And here is a diagrammatic picture of a lab that we were able to set up for the Nelson Mandela Institute for Science and Technology in Arusha, in Tanzania, where one of our colleagues had developed a project looking at emerging pathogens in bushmeat. So here we developed a diagnostic lab for them, a molecular diagnostic lab for them. And this is how the layout of the lab is. Very simple, easy to construct, and easy to maintain. It all starts off with the sample coming in to the receiving and accessing room, followed by DNA extraction, and then moving into the first area of the clean area. And then we have the master mix that comes from an external source, which is mixed and then taken into the PCR room. So what about the post-capture and analysis of data? When we look at post-analytic, is one of the foremost aspects of a quality assurance and quality control is minimizing the efforts made to actually physically write the data. It would be great if you can take all of the data that comes out from your PCR machine and gets incorporated directly into the laboratory information system. So there are a lot of errors that can be induced by virtually doing manual entry does get eliminated in the process, okay? The next is standard operating procedures for integrating results into the master database, validation, review of Levy Jennings charts, and I'll talk about that in a slide or two. And then finally, it is reporting and invoicing. How do you report to your client? Is it just sent to the client or is it sent to the veterinarian? And more importantly, this is where the success of a diagnostic lab comes, is the case coordinator, the person who is handling the case, is available to talk to the client and the veterinarian and discuss their results of analysis. This, I believe, is a very important part that some of the diagnostic labs don't do a very good job, because this is the way that we build a trust between the diagnostic lab and the clientele, so that we keep them engaged and they come back to us and utilize our services. So here is an example of a Levy Jennings chart. And this Levy Jennings chart shows us here the readings on the y-axis and the number of days through which these data points have been captured. Here is a data for a PCR-based assay, where we are looking at changes in the concentration, ionic concentrations within 1 standard deviation. And for us, the limit is anything that exceeds 2 standard deviation means we are having an issue with our assays. So you can see just around day 30, there were 2 separate instances in which the controls did go out of the 2 standard deviation. So as a result, an intervention was made to see, do we change the reagents? Do we come in with a new set of controls? And when that particular process was put into place, you can see the cycling conditions or the PCR assays or the chemistries have fallen back within the 2 standard deviation. So this becomes very, very important to build in laboratory quality control based assays, is not only to assure yourself, but also to assure your clients that the quality of the data, the quality of the results that you're giving them is of utmost quality. So coming back to the question, what went wrong with Dr. Sharma's PCR? I would have loved to enter into a chat discussion, but for sake of time, I will kind of go on a limbo here. So what really happened? When we look at Dr. Sharma's PCR test, they didn't really spend any time looking at what should be the components of the pre-analytic. How should the samples be collected? How should it be transported? Once it's received, how do we go and work with it? The next is post-analytic. We did see that there were a lot of complaints from the clients or questions from the client, how to interpret the results. And that really boils down to very much to the analytic phase of the continuum, where they did not really do any validation. They did not get any field samples. They did not get any field-based isolates. They did not do any blind testing. There were no references to quality assurance practices or quality control practices that were followed in the lab. So we can see Dr. Sharma did not have a strategy, nor did he have any tactics. So here, the lesson learned is, before you develop a diagnostic test for your clientele, you need to focus on the pre-analytic part, the analytic part, and the post-analytic part so that they are seamlessly integrated with each other. And the test that you offer complements the three different parts of the continuum in order to offer the best available test. Okay. With that, let's move on to the second act. Who should lead, whether it's a pawn or the horse? What's your choice? So let's start off with a little bit of where we are as it refers to bacterial identification systems. So the platforms that are available for this part of the identification system is we can use phenotype-based platforms, genotype, antibody, or chemotaxonomic-based systems. So when we talk about phenotype, they're largely done employing biochemical tests. And using biochemical tests, phenotype-based assays allow us to identify the family, the genus, and the species, but are unable to help us differentiate the species, the strains within a given species. So that's one of the limitations. The other one that is complementary or very similar to the phenotype-based in terms of the output of information that we generate is for example, MALDI. MALDI also gives us the same kind of information as phenotype-based assays do us. Family, genus, species, and that's it. So here, either you have an option, you go with the phenotype-based assays or a chemotactic-based assays in order to incorporate that as a part of your diagnostic laboratory system. And somewhere in the late 2000, early 2000, I would say, antibody-based assays were also developed largely to identify species, bacterial species, or more importantly, different viral entities. We could do that very effectively, especially in a high throughput, high volume lab, like in a hospital setting. You would like to screen for example, cytomegalovirus, or you're looking for HIV or other things. That's where the serology-based, antibody-based assays became very, very useful and powerful. But when we look at the last one in the whole set of these four different platforms, it's the genotype-based assays. When we look at genotype-based assays, they comprise of RT-PCR, DNA sequencing, and RNA sequencing-based assays. So when we start off with RNA-based sequencing assays, it is pretty similar to the chemotactic and the phenotype, where it allows us to identify the family, the genus, and the species. Whereas RT-PCR-based assays allow us to identify the species and also allow us to do some strain differentiation, and more importantly, helps us to identify specific virulence factors in addition to identifying the species. But the big kahuna in the whole genotyping-based system is using sequencing-based systems that allow us not only to identify the family, the genus, the species, but also identify or differentiate the strains within a given species. So you can see here, I've laid out a ground here for us to move forward and talk about some of these platforms that we will be looking into the next set of slides. So for those of us who studied microbiology in the early 80s, or studied microbiology, and I would say in the late 70s, the most valuable book in any lab that does bacteriology work was the systematic bacteriology one. And in fact, I still recollect my professor of public health, Dr. Koth, would keep these manuals under lock and key. And whenever we needed it, we need to go and request him to allow us to use this book. So this is a fascinating book. And even today, the amount of information it contains is just astounding. So when we look at the Burgess manual, the first one is what we call as the systematic bacteriology, is a more recent version, which incorporates the evolutionary and the genetic relationships when it comes to classifying bacteria. Whereas the very original one, that is the determinative bacteriology, is just exclusively based on classification by cell wall, composition, morphology, biochemical test, and different staining characteristics. So let's talk about phenotype-based identification systems. So let's use a timeline or a continuum between 1900s to the end of 2000. So sometime around 1900, the way microbiologists or people working in a diagnostic lab would identify bacteria in the 1900s was exclusively based on morphology. How does the colony look like? Is it dry? Is it moist? What is the color? What are the margins of the colony? The next around 20 to 30s, they started incorporating cultural characteristics. Does it grow in a broth? Or does it grow with a change in color? Does it become more acidic? So they talk about cultural characteristics. And somewhere between 1940s and 50s came in the concept of utilizing biochemical tests, followed by between 60s and 70s, utilizing enzymatic-based assays to look at susceptibility patterns. And then finally, late sometime around the 80s and 90s, we came across the process of also identifying some virulence factors. So the assays that were developed in the 40s and 50s, around 60s, 70s, were called as conventional methods. Now, these conventional methods were labor intensive, took up a lot of laboratory space, took up a lot of time, especially to prepare the media, and more importantly is the interpretation of the results. Now, in order to overcome these hindrances, scientists developed rapid and miniaturized identification systems, such as the APIs, the biologs, the sensitizers, the YT2 compact systems. So somewhere around the 80s and the 90s, the conventional testing methods became obsolete and were largely replaced by rapid identification methods. The other one that we have seen a big increase in the number of applications as it relates to antibody-based identification systems that came in sometime around 2000 and continued all the way probably till 2020. These systems were, as I mentioned earlier, were utilized in hospitals or in a large diagnostic lab or a centralized testing facility where they could run several hundred samples at one given time. Again, these were limited to a certain set of species, more so for viruses or highly pathogenic strains of bacteria such as E. coli 157. So there were pathogen-specific based assays when it comes to antibody-based identification systems. The next one is the proteomics-based identification systems. We started seeing the very first prototypes emerge sometime around 2010. And sometime by around 2015, we started seeing the development of benchtop-based ID systems have become, and these systems have become virtually the workhorse in many diagnostic labs, at least across the US and Canada. MALDI-TOF has replaced the rapid identification systems and other phenotype-based identification systems in a diagnostic setting. So the last one which I would like to talk about is the genotype-based identification systems. So when we look at genotype-based identification systems, the use of 16S rRNA became very popular in the 1990s, because that was when we were able to sequence a 1500 base pair using 3 different gels that would cover us anywhere up to 300 base pairs. So that allowed us to identify the bacteria based on the 16S rRNA-based sequences. Sometime around 2000, where PCRs became the regular run-of-the-mill. It's like having a pressure cooker at home. So PCR-based assays became virtually an absolute essential instrument in a diagnostic setting. So using PCR-based assays, several different assays were developed. And many of them did really work very well for doing disease diagnosis and identification of pathogens. But 10 years later, sometime around 2010, was where we started seeing an explosion in the use or the application of whole genome sequencing, as many different systems were developed, such as the Illumina-based, the Ion Torrent, and of course, along with it came kits that you could utilize in order to identify the species of bacteria based on gene sequencing. So what are some of the limitations we see when it comes to phenotype-based methods? Some of the limitations are, one is you need to cultivate. You need to cultivate the bacteria or the pathogen on a petri dish. You must have pure cultures. And one of the drawbacks that we tend to see with phenotype-based methods is highly related species, that means those that are fairly conserved with very little divergence, we find it very difficult to differentiate them using phenotype-based assays. And many a times, in order to use phenotype bases for identification, takes anywhere between 72 to 96 hours. So let's move on to the third act. Think before you move. Phenotypes or genotypes or both. Let's enter into the new millennium, DNA and RNA-based assays. What we know, they are more accurate, they take less time, but of course, the big question is always there: how much is it going to cost us? Yes, we do know that the cost of doing nucleic acid-based assays has significantly dropped as compared to the last 5 or 10 years, and we hope the Murphy's laws will still continue play to work here, and hopefully we should be able to see these costs driven down significantly in the next couple of years. So the question that I would like to pose to all of you is, why are we so excited about using DNA-based assays? The reason is techniques are now available that permit analysis of the whole genome to the extent of finding a difference in one nucleotide within and between genomes. You can see the power that you have with this technology. The next is DNA-based methods allow discrimination of strains that are indistinguishable based on their biochemical or serological tests. So what biochemical and serological tests can't do for us, DNA-based assays can step in and help us sort out the different strains. So looking back in the last 15 years, genotype-based methods have proven to be very beneficial in overcoming some of the limitations that we have seen with phenotype-based assays for bacterial identification systems. So when we look at genotype-based assays, we start off with the basic presumption is genotypes of every individual is unique. And we can look at the target either on a single locus or multi-locus, or we can focus on the whole genome. The assays tend to be sensitive and specific. The speed of the assay can be as less than 3 hours to the extent of 96 hours if you are doing a sequencing-based approach. One of the fascinating aspects of genotype-based assays is we can directly probe into the specimen itself without actually enriching for the pathogen or growing the pathogen. So we can directly go to the specimen, extract our nucleic acids, and start doing the assay. But however, keep in mind, they require extensive validation. And many of the assays that we see in the market and are currently driving the molecular diagnostics are PCR assays, nested PCRs, multiplex PCRs, RT-PCR-based assays, oligonucleotide probes, and of course, the whole genome sequencing. So people typically tend to ask, so what is the gold standard? Yes, you have developed an identification method using a certain platform. What do I compare this with? So you compare this with the 16S rDNA sequence analysis, because that is considered to be the gold standard. And how do we do this? We know for sure the DNA coding of 16 RNA is present in all bacterial species. There may be multiple copies, as low as 5 copies to as high as 20 copies. And when we look at the 16S rDNA, they contain fully conserved and variable regions. And what do? We target the conserved regions in order to identify and amplify the particular portion and then sequence that variable region that will allow us to identify the species. Here in my next slide, I've kind of laid out where do we focus in terms of doing the amplification. I will not go much into details, but this is pretty standard. We amplify regions V1 to V2, V3, and V4, add in the barcode sequences, clean up the PCR prep, run it on a sequencing platform, and then do the data analysis. Of course, real-time PCR, as I mentioned earlier, is like having a kitchen sink with you nowadays. It's highly sensitive, specific, and it has a fantastic dynamic range. And that's what really excites me. Next is it is simple, rapid, and reproducible because many of these you can buy off the shelf and start doing some of these assays. They provide real-time detection and quantitation, and especially gives us an information about the pathogen load. What is the likelihood of the pathogen in the given sample? There is low risk of contamination. We can automate the system. And of course, over time, the chemistries have become very reliable, and we can develop targeted pathogen-based detection and typing systems. So let's move on to the one which I would like to spend a little bit more and talk in my next set of slides is the DNA-based sequence methods. When we look at DNA sequence-based methods, the sequence data is unequivocal. So that's why we say this becomes a reference standard. We are seeing over time there is improved reliability and reproducibility of the information that we generate. More importantly for me, it's easy to compare with other laboratories. If I sequence a virus, if I sequence a bacteria, I can send in the sequence data to another lab or another colleague of mine to compare or analyze. More importantly, we don't really have to distinguish between culturable and non-culturable organisms. The public databases are increasing in their size, or exponentially increasing in size. The assays tend to be fast, accurate, and the costs are coming down. Not to the extent where we are, but we definitely would like to see the cost come down a little bit more. But the take-home message as of today is these technologies remain rather complex, and successful adaptation of these in a microbiology lab will require expertise in molecular biology and bioinformatics. Okay. So here is another picture where I kind of like to share with you is we tend to focus on the genome, but the ultimate for me or other people in a clinical setting. When we are looking in a clinical setting, you have a patient, you go into a farm, you have a flock of birds or you have calves in a pen, and you would like to get a whole holistic picture of what's going on. I would say the ultimate diagnostic tool would be used to provide a practitioner with not only the genomic data, but also the transcriptome, the proteome, and the metabolome data that can be given at the right time in the right kind of approach so that they can make practical decisions in order to develop treatment or intervention protocols. Okay. So when we look at whole genome-based sequence or sequence-based approach, we generally like to apply an unbiased based sequencing approach, whether we use the whole genome sequencing or whether we take the metagenomes-based approach to discover a pathogen. So here is a slide, I'll not spend much time here, is largely kind of depicts the evolution of the sequencing technology. The very first one started off with the Sanger sequencing in 1977, started off with gel-based assays, creating acrylamide gels, putting it between two glass slides and praying that the glass slide doesn't crack when you pour in the gel, and you monitor the temperature of the glass so that it doesn't start leaking on the sides. Been there, done that. So good riddance gel-based systems, because I couldn't get more than 200 base pair reading on a good day. If it was a good day. So that's where we all started, was Sanger sequencing in 77. Of course, followed by the next generation, the 454 revolutionized the way we look at genomes. of course, then we have the Illumina-based platforms and the iron torrent-based platforms that have kind of revolutionized, and we are moving so fast in terms of applications and different modalities when it comes to sequencing. I wouldn't be surprised we have already crossed the threshold of the 4th generation and already moved into the 5th at this point in time. Okay, so here is a kind of a layout how high throughput sequencing is done. I will not go through much of it because I know my colleague Dr. Suresh is on the other side, so I'll quickly try and wrap up in the next set of few slides. So in summary, when we look at high throughput sequencing, it offers a lot of opportunities to identify several multifactorial diseases and co-infections, diseases of unknown etiology, vector organisms and reservoirs, new and variant divert pathogens, and more importantly, it allows us to improve our diagnosis as it relates to emerging or re-emerging diseases with known and unknown etiologies. But there are some challenges that still emerge. High throughput sequences generates large amounts of data, and that is one of the issues that we have issues with. Another is, how do you store and do the bioinformatic analysis? How do you validate different sequencing approaches for diagnostic use? So, Dr. Suresh, probably he will talk a little bit about validating sequences for diagnostic approach, probably in his presentation. Okay, guys, I'm not able to move my slide. Okay. Hello.
Dr. Mote, please make your mic off. Dr. Mote, please.
Thank you.
Sorry, please continue.
Okay, let's move on to the 4th act. Antaryami, that means the ever-present, the omnipotent, the genes and the genomes tell it all. So let's go and talk about what have we done. So Dr. Suresh Kuchipudi and myself, we have developed a new section within the Penn State Animal Diagnostic Lab. We call it as the Genomics and Bioinformatics section, largely to develop applications that we can use in veterinary diagnostic medicine. And now let me share with you some of the services that we offer based on whole genome sequencing. So the whole genome sequencing that we offer to the Penn State Animal Diagnostic Lab, if you send us an E. coli sample or a salmonella or any bacteria, if you send us and would like us to provide us with you a pathogenomic analysis of the organism, what do we typically provide you with? We provide you with a serotype, especially if it is an E. coli or a salmonella. We'll do multi-locus sequence type, especially if it is MLST or core genome MLST. And this is very important largely to allow tracing within a flock or a herd or between herds in a county, state, or at a national level. We give you information about the antimicrobial resistance genes, the genes that encode for virulence and mobile genetic elements. And this is the report that we typically generate and share with our clientele. It kind of starts off with telling us in terms of person, place, time, species, age, sex, and location. What are we looking at? What is the history? And what were the interpretation of the findings? Remember, many of our clientele have very little time. If they have 1 to 3 minutes, especially a practicing veterinarian or a physician, they have very little time. You need to capture the information in 4 to 5 lines that will allow them to make a decision or move them to the next level to make the appropriate decision. We then provide a comprehensive listing of virulence factors, antimicrobial resistant genes, and a kind of in history where this particular serotype and sequence type has been previously identified. Okay. So where would it be helpful in practice? So when we look at it in terms of a practicing veterinarian, comprehensive analysis of virulence factors, why are we seeing the onslaught of the same type of pathogen with the same type of clinical conditions when you want to serotype E. coli or salmonella? More importantly, from an epidemiology point of view, can we use WGS-based data to troubleshoot persistent and recurrent infectious disease problems, okay? Address multiple outbreaks in your practice. And the last is to characterize the pathogen before making an autogenous vaccine. So what is the cost? Ultimately, it comes down to how much is it going to cost. When we look at whole bacterial genome sequencing, the cost that we do is about $120 for an isolate. That includes the $25 for isolation and identification directly from the sample, plus doing the whole genome sequence analysis. As compared to doing conventional tests, where we also will require to do virulence typing and serotyping. And when we employ conventional typing, it will cost us anywhere from $75 to $115. So it is comparable. WGS is almost there. Probably in a year or two, we may be able to do much better in offering a more comprehensive WGS-based analysis. The last one, checkmate, okay? I'm almost done. So let me share with you a project that we have started where we are looking at metagenome, looking at developing a metagenome-based surveillance system to determine the prevalence and distribution of known, emerging, and previously unknown pathogens in commercial backyard and wildlife birds. Okay? So let me kind of introduce you to the word metagenomics. The metagenomics is the study of bacteria or viral DNA extracted directly from the biological samples, from the biologic samples. So in coming years, we definitely know sequencing-based technologies will most likely replace some of the conventional pathogen identification techniques, such as bacterial culture, virus isolation, PCR, and immunoassays. Those of us who are traditionalists really fear that we were going to lose this vital part, but this is just with some of the assays, okay? And many of the systems that we use are currently used for surveillance of infectious diseases. So here is a kind of a layout how we do the next generation sequencing. There are several NGS approaches for detecting pathogens that are of importance to human, animal, and human health. So sequence-based directed NGS helps us to know what you're really looking for. And the process is very simple. You extract the viral RNA or DNA. You prepare a genomic library by fragmentation of the RNA or the DNA, attach a probe, what you're really looking for, and then you extract the hybridized probes with the help of a magnetic bead and subject it to whole genome, subject it to sequencing, collect the data, do the bioinformatic analysis, and that will allow you to identify the targeted viruses or bacteria. Okay, what are the strengths? We'll identify known pathogens and the technique is sensitive and specific. And what are some of the limitations? Unable to identify novel and newly emerging pathogens. So how do we overcome this limitation? So there's a new technique that has a lot of promise, sequence independent single primer amplification, or called as Cispa, for detection of new and emerging infectious disease. In fact, I should compliment a group, I believe in Gujarat, from Anand, did a very good project on Cispa looking at poultry viruses. Was very much impressed with the study they had done. So here, the Cispa project that we would like to do will focus on sequence-independent next-generation sequencing. You know what you're looking for. In addition to that, we can also detect pathogens that we have never seen in a given herd, flock, or a given community. And how does this, how have we planned this study? We plan to collect oropharyngeal swabs from chicken. It may be bacteria, viruses from the oropharyngeal area, extract RNA or DNA, selective remove the chicken DNA, enrich the RNA and DNA of these microorganisms, and then perform sequence-independent single primer amplification, wherein we convert the viral RNA with the 8-mer-based viral DNA adapters to it, do the cDNA amplification, and then subject it to sequencing. So this process will allow us to not only identify the known, but also the known likely and the emerging pathogens. Okay, so this is largely called as Cispa NGS. What is the value of this tool? It can be used to detect viruses and emerging pathogens at ports of entry, imported food products, travelers suggestive of viral or bacterial infections at international airports, illegally bought products and animals, bushmeat, endangered species, birds. More importantly, regulatory agencies will be able to use this to monitor poultry, dairy, captive deer, wildlife, aquaculture, especially at hotspots such as fairs, live birds, and auction markets, more as a preemptive strategy for detecting newly introduced pathogens that have not been seen previously in a given area. So in summary, I think I have sold you an ultimate tool that you can do everything with one single system: species, serotype, genotype, AMR, virulence factors, and mobile elements. Having said that, it largely will depend upon what you're really looking for, okay? Yes, WGS, next generation sequencing, is promising, but we still have a long way to go, especially when it relates to the robustness of the pre-analytic segment, especially when it comes to metagenomes. If your sample is not collected properly, if it is not stored properly, your metagenome data will significantly scream back at you and say, you're not doing things right. So this is one of the areas that we need to spend a lot of time, especially when it comes to applying Cispa NGS. Better flow through analytics and lower analytic costs. Can we decrease the cost of the reagents? Can we develop customer designed reports? Are some of the challenges that we will face when it comes to applying WGS or NGS. So my last slide here is, this is the way where we see genomics and other omics-based sciences drive diagnostics. Not only diagnostics, but also help us in doing surveillance, develop vaccines, and that's what Dr. Kuchiburi will be talking about. And of course, also developing the future therapeutics. With that, I will stop here. And if you have any questions, I will try and answer now, or probably perhaps after Suresh finishes, we can all be there to answer any questions if they are there. Thank you.
Thank you. Thank you, sir.
Thank
Very
you.
exhaustive, very informative talk. Too many topics in that talk, and we are really fortunate to have your talk today, and we are really fortunate to listen to the next talk also. Shall we take questions now or later on? And I request the participants to put their questions in a chat box, so that my co-chairman will pick up those questions and ask to the speakers. So, sir, Jairav sir, whether we should take it now or later?
Let's do one thing. It would be great if Suresh finishes his, and then he and I can stay back in the end and answer any questions. Are you okay with that, Suresh?
Sure, Bhushan. Either way, I'm fine. Either you can take the questions now or after my talk.
Dr. Kurkure, I'll leave it to you.
Okay. So,
I only see one question in the chat, so maybe you can just address that and then we can move on.
Mobile, the question is, what are the mobile genes, mobile gene TIC element, transferable element, and what is the function of these elements in gene sequencing?
So many a times, it's a good question, very good question. What we are learning is more and more is many of the mobile genetic elements that we put them in a basket that comprises of transposons, integrons, plasmids, or phages, prophages, whatever you're talking about. These are known to encode for virulence factors, some of the phenotype related characteristics. They may encode for resistance to antimicrobials. And many a times, if we don't look into the mobile genetic elements, we will be not in a good position to answer the question. We have two isolates that have the same identical multi-locus sequence type. They have the same serotype, but why is it that their antimicrobial profiles are different? And then when you look into the repertoire of the mobile genetic elements between these two organisms, we start seeing the stark difference of the absence or the presence of a mobile genetic element or a plasmid that it may have acquired in that very short period of time, and thereby become resistant to a few more antibiotics than its parent strain or its related strain, right? So that's where the mobile genetic elements gives us a more refined picture on the molecular or the molecular epidemiology picture that we're trying to paint with the given pathogen.
Thank you, sir. Another question is how you design a primer for Cispa, since we don't know about new pathogens or having any previous database for the new pathogen.
So what the concept is a little different here when it comes to Cispa. Is what you do is you take Kenlo fragments. The Kenlo fragment can be a 6-mer or an 8-mer, which has a poly A or a poly T tail that will link on to certain aspects of the genome, and we will amplify it. Okay, it is normally present. So once we amplify the Kenlo fragments, we can then clean up the Kenlo fragments and then apply it for sequencing-based processes. That's how we do. So we don't really look for any specific target. We can do that too, but here is amplifying what we know, remove that that is not a part of the organism, or remove the host interference, and that will allow us to do a better job.
Thank you.
Sure.
One very interesting question. Do phenotype and genotype always correlate? If not, what must be concluded? Should the result of genotype or phenotype be considered?
I'm sure Suresh and I had talked yesterday. It's the chicken or the egg story here, right? So if I were to be given the opportunity, is what is it that can give me the best information, the quality of information, more important, the quality and the accuracy of the information for the time that I'm investing and the resources I'm investing, right? So if you don't have a laboratory that is adequately set up to do genotype-based assays, your best option is to go with phenotype-based assays to give you the information that you're looking for. So largely, I would say is depends on the laboratory, the access to some of these different platforms in order to identify the different bacteria. And many a times in a given setting, like in our diagnostic lab, we like to get a little bit of an understanding of the phenotype, not the complete process. For example, if I'm growing E. coli, I see, sorry, let me take back. If I'm seeing growth on a McConkey's agar, and I see the presence of pink color with a halo around it, my initial assumption it's E. coli. Does not necessarily have to be, but at least it drives me to make a better differential diagnosis so that I can apply the right kind of genome or nucleic acid or a phenotype-based assays to answer that question.
Thank you, sir. Thanks for the answers. And in chat box,
can I just add, if you don't mind, can you hear me?
Yeah, please.
Yeah, just to add to the question, this is such a fascinating question in genetics. So in the spirit of the question, so do phenotype and genotype always correlate? The simple answer is no, because of what we understand about genetics. But the question is in the context of Dr. Jayaral's talk of the choice of diagnostic tests that you use, whether it is phenotype-based or genotype-based. It depends on really what we are trying to understand. So the presence of a gene or a mutation doesn't necessarily mean that it translates into something functional. So it really depends on the intention and what you really want to do. So if you want to find out, for example, antibiotic sensitivity patterns of a bacterial pathogen or others, And there are perhaps more phenotypic-based methods are more relevant. But there are sometimes you want to distinguish minute differences between bacteria that may not show phenotypically, but they do have variability in their certain parts of the genome that could be relevant for vaccine. So the question is then, it depends on the context, and more importantly, it depends on what you really want to accomplish.
Thank you, Dr. Suresh, for adding value to the answer of the question. I could see a lot of appreciation in the chat box about the talk. So I thank Dr. Jairao sir for a very nice, informative, and very nice talk. So now there are some questions.
Submission.
We will take it later on. So now I request Dr. Sonal to please introduce our second speaker. Dr. Sonal, can you listen me?
Yes, sir. Yes. Sir, am I audible?
Yeah, sure.
Just trying to share my slide.
I don't hear any audio. Maybe it's just me or everybody else.
No, sir. They just.
Query, can I tell, please?
Okay, so again, thank you, Jairao sir, for your informative talk. Now I again take the opportunity to introduce Dr. Suresh Kuchipudi sir, who is currently working as the clinical professor and head of microbiology section. He's also the associate director at Animal Diagnostics Laboratory at Huck Life Sciences, University Park. Sir has completed his bachelor's from Acharya N.G. Ranga Agricultural University, Hyderabad, and he has done his master's from Tanuvas. Followed by that, he pursued his PhD at the University of Glasgow and postgraduate certificate in higher education. Sir has also been certified by the Board of American College of Veterinary Microbiologists as a specialist in virology and immunology. His areas of expertise are infectious diseases, pathogen-host interactions, zoonotic and emerging viruses, vaccines and therapeutic development. He also develops diagnostic assays and also viral pathogenesis. Besides, again, he mostly works on his research is basically based on the investigation of host and viral determinants of zoonotic virus spillover, mechanisms underlying the emergence of new epidemic or pandemic due to viral pathogens. As far as I understand, sir is also involved with certain studies with the now ongoing pandemic of COVID-19. And he's also into the development of innovative diagnostics. And his laboratory is currently studying the wide range of emerging and zoonotic viruses, including the COVID-19 virus. One of the major achievements, he has generated funds worth $10 million US from National Institute of Health, National Science Foundation, and USDA National Institute of Food and Agriculture. He has also 66 publications at his credit. And as Kurkure sir had earlier mentioned, it was really difficult to summarize the achievements of Dr. Jairao sir also and also Dr. Kuchipudi in one single slide. So I'm sorry, sir, if I've missed something, but we welcome you. And the speakers are, the participants are really eager to listen to your talk. And I hand over the session again to Dr. Kurkure sir and Dr. Deshmukh sir.
Thank Dr. Sonal for a nice introduction about Dr. Suresh. And again, I will not be in between the speaker and the listeners. So over to Suresh. Dr. Suresh, please.
Thank you. So first of all, I would like to thank the organizers, Dr. Kurkure, Dr. Zende, for the invitation. It's a pleasure to participate in this training. And also, thank you very much, Dr. Sonal, for the nice introduction. So let me bring up my slides. Can you see my screen?
Yes, yes. Only thing, yeah, it's better. Yeah, good.
Yeah, thank you. So as Dr. Jairao has outlined the importance of next generation sequencing and how it revolutionized the practice of veterinary medicine. For this, the purpose of this talk, I'm going to focus on some specific developments in the vaccine technologies and how they're relevant and applicable in the veterinary medicine field. So before I start talking about this, I wanted to show this slide just to emphasize how unprecedented times we are currently in. But this is the snapshot of COVID-19 map from Johns Hopkins University in the US that just shows as of 8:30 a.m. in the Eastern time, just an hour ago, the cumulative cases and the deaths globally caused by the ongoing COVID-19 pandemic. So there are more than 3.5 million deaths and still counting. And I also wanted to show you this picture if anyone can really figure out what this picture actually is showing. I believe you can't really unmute yourself and talk, but if anybody recognizes what this is, what this might be, you can put it in the chat window. I'll give a few more seconds. Vikas says plastic waste, dead bodies, clothes. Anything more specific? Anyone? Dead animals, that's close. Balakrishna says. Okay, that's good. So you're guessing what it is. So this is the aerial view of a mass burial of cattle during a foot and mouth disease outbreak in Japan. And it really shows the extent of the devastation that an emerging infectious disease or a previously known, but an outbreak of a deadly viral pathogen could cause. So the point I just wanted to highlight is that infectious disease outbreaks, whether they are human or animal related disease, they could cause a devastating impact on animal and human health. And in fact, we all have experienced and currently living the far-reaching implications of a pandemic. It's just not about the health, but it just completely changes the life as we know. So, and with that, the point is that there are interventions that could help us, and one of them is vaccines. And we all have learned how quickly some amazing vaccines have been developed. So what I'm hoping to do this evening is to provide you a brief overview of the classical versus next generation vaccinology. What is the approach and what are the differences? And also introduce to you to the concept of reverse vaccinology, and then specifically focus on three vaccine technologies: the viral vector vaccines, DNA vaccines, and mRNA vaccines. So before I move on with the topic, just wanted to introduce my lab and a whole bunch of wonderful people that work in the lab that do all the science in the lab. I am part of the Center for Infectious Disease Dynamics at Penn State and also the Center for Molecular Immunology and Infectious Disease. And I'm a member of the Molecular, Cellular, and Integrative Bioscience graduate program and also the Pathobiology graduate program. And we study a range of emerging viruses of both animal and human health importance. And the particular focus is on viruses that have the zoonotic potential, including the SARS coronavirus 2, which we have been researching for over a year and a half now. So I'm sure this audience know very well, but just to introduce the concept of a vaccine. So you all know, you all have learned that a vaccine is a biological preparation that provides active acquired immunity against a certain disease. And it is really critical to understand this is active acquired immunity because it is not natural. And you always hear an argument from people that that sometimes try to be dismissive that it may be better to naturally expose to a disease and become immune, and taking a vaccine is artificial and hence it is not really good. So that is the ideology of anti-vaccine sentiment. But as veterinarians and scientists, we know that this is the safest way to become immune and potentially protect from a deadly disease that could essentially has an opportunity to kill us. Okay. So vaccination saves lives, as we know. I did not mean to put this arrow here. How do I get rid of this? Never mind. Okay, so we know that the vaccination saves lives, and it has significantly helped in reducing the devastating impacts of endemic viral infections or emerging viral infections of animals and humans. And we also know how the COVID-19 vaccines have been playing a significant role in protecting people and potentially bringing this pandemic to an end. But more importantly, we also must recognize and remember that vaccination also helped us in the successful eradication of some deadly viral diseases. To name two most famous examples is smallpox in people and the rinderpest virus in animals. While we don't have many such success stories for a number of different reasons, but the technology continues to develop and there is a lot of hope. So when we focus on the veterinary vaccines, the classical vaccinology approach that we all know and is still being used, and of course it's valuable. It has a pipeline that begins with isolation of a pathogen. So whether it is bacteria or virus, we isolate the bacteria or the virus. And then we take the pathogen through a process of attenuation. So you basically try to weaken the pathogen or inactivate it so that it is no longer able to cause the disease, meaning there is no pathogenicity, but you retain the immunogenicity, meaning it is still able to inform the immune system to respond. And it also stimulates the memory. So basically, you accomplish the purpose of an immune response to protection without the adverse effects of the disease. And there are several subsequent iterations of this approach have come moderate to maybe perhaps a bigger advances in this. Is where we can purify some antigenic factors of the whole organism. Rather than using the entire organism, you could purify parts of those bacteria or viruses and could be used as vaccines. But one of the major limitations with this approach is it is a very laborious approach, and it also yields few immunogens over a very protracted time frames. And especially if you are in the middle of an outbreak or a situation where you have to vaccinate millions of animals, then this approach would not give you the necessary amount of vaccine that is required to vaccinate. So then what is the alternative? So we have made a significant progress from the time Edward Jenner did this vaccine. We all have learned in the vet school the history of vaccines. And from this time on, the science in general has made a tremendous progress in advances in vaccinology. And now we are talking about the new generation or the next generation vaccinology. So as Dr. Jayarav very nicely laid out, all the different possibilities that the sequencing or the next generation sequencing has given us also is to transform the way we develop vaccines or the way we design vaccines. So the new generation or the next generation vaccinology essentially involves to predict a vaccine antigen computationally from the DNA sequence of pathogen. So without even growing the pathogen or learning much about it, we can computationally predict what are the antigens that could be used. And then from these proteins, then we can select those that can specifically or efficiently activate both arms of immune response, the T cell response and the B cell response that is important especially for protection against a viral infection. And these can be predicted in silico, meaning you analyze the sequence and you can predict them. And along with this methodologies, we also have made significant advances in understanding the pathogenesis of several pathogens. And one of the key aspects is to know what are the correlates of protection. So for example, currently we are learning that with SARS coronavirus 2, the correlates of protection are most likely be the neutralizing antibodies. So we need to understand what would be necessary to happen in a recipient of the vaccine, be it an animal or a human being, to establish that they do have the protective immune response without having to challenge in an experimental setting like what we do in the lab. So the concept of reverse vaccinology, it really was something pioneered by a scientist by name Rna Rapoli. And he first used this approach to design vaccines against the serogroup B meningococcus. And this is now being extensively used for several other bacterial vaccines. And this is indeed a significant progress from the time Louis Pasteur made his vaccines. And it also relies heavily on using the genomic information and using the advanced bioinformatics tools. So how do you do this? You first sequence the pathogen, and the sequence analysis would help us to identify specific genes. And then we identify the specific genes that could be of antigenic value. And then you clone the gene of interest and create a recombinant construct of the gene. And then from that, you could take a number of approaches. You could then make a DNA vaccine that I'll be talking about briefly. Or you could design a subunit vaccine, or you could put it into a viral vector and make a vector vaccines. But regardless of what would be the end platform that you will use, the approach will be starting from the gene sequence and then computationally identifying what antigen should be used for the vaccine. So it is also important for us in the veterinary field to recognize that how complex our clients are, our patients are, because in the human medicine, you want to learn about the way a human body reacts to a vaccine, and then the set of pathogens that are relevant to human disease, and the vaccines are designed, and sometimes people just understand, or sometimes they just simplify the fact that, oh, there is a vaccine for a certain disease in people. Why can't you just use that in cattle, for example, or dogs? And the answer is, it's not that simple, as we all know, because we have several species that we want to consider, and their immune system is very different. And we need to fully understand or take into account the differences in the humoral and cellular immunity of these species. For example, the way you design a vaccine for chicken is very different from the way you design a vaccine for a dog, for example. And in terms of the vaccine itself, we need to think about how are we going to deliver the vaccine? What is the route of deployment? And what antigens we need to use? Are we going to use a whole inactivated pathogen, or you want to use a live attenuated vaccines? And there are considerations for using some of them in certain settings, and some of them cannot be used. For example, if you have an RNA virus like SARS coronavirus 2 or influenza virus, for example, that mutate, and it is not a safe approach to use a live attenuated vaccine, because the vaccine virus could mutate and revert to pathogenicity. And similarly, you could also use the recombinant vectors or various different things. So we need to make a conscious choice about what antigen type we are going to use. And then based on the type of antigen, we need to determine if there is a need for an adjuvant. So there are a lot of considerations to make before you design a vaccine for veterinary application. And the point here is to emphasize that there is no one size fits all that works in the veterinary field. So type of viral vaccines, if you look at them, there are a whole bunch of them. And in the interest of time and the context of this training, I decided to focus only on the nucleic acid-based vaccines, DNA, RNA vaccines, and the viral vectors. But I also wanted to highlight the fact that there are a whole bunch of other ways in which you can make a vaccine. For example, the conventional live attenuated whole inactivated vaccine, or you could use a synthetic peptide, and VLPs are becoming popular, virus-like particles, or you could do a recombinant subunit viral protein. So again, I apologize if it is becoming too technical. I recognize that there is a broader audience, so I try to keep it really simple. So conventionally, we were using the entire organism, whole inactivated, as a vaccine, or take parts of the viral proteins as a subunit as a vaccine, or you attenuate the difference between inactivated and attenuated. I'm sure you all know is the attenuated vaccine, the virus is still alive, but you transformed it in a way that it is no longer able to cause the disease. Whereas inactivated virus vaccines are killed vaccines that are no longer able to cause any disease or replicate, but they retain their architecture. And similarly, the virus-like particles are basically a shell that is decorated with the peptides that are immunologically relevant. So for example, if you want to create a virus-like particle for SARS coronavirus 2, so you would decorate, say, a lentivirus or a vesicular stomatitis virus, some backbone, not the coronavirus, but then we will decorate it with the spike proteins from the coronavirus. So your immune system will recognize these proteins and produce antibodies against these particular proteins. And the most exciting things in the field are the next generation vaccine platforms, which I'm interested in and we'll be talking about today. They use nucleic acid based methods, and they really represent an approach where synthetic sequences could be used to express antigenic peptides or proteins in situ. And if you think about the vaccines that really made a big difference in this pandemic, and that is an incredible progress we made in the vaccinology, that in less than a year after a global pandemic with a completely new virus, there are more than 5 very effective and safe vaccines have been produced. So this is all the recognition of the advances in science we've been making over the past decades. And some of them are RNA-based vaccines, and some of them are viral vector-based vaccines. So I'll be briefly talking about viral vector vaccines, DNA vaccines, and RNA vaccines. Before I move any further, I would also want to periodically check in the chat box. If you do have any questions, feel free to put them in the chat and I can address them as I go along. So the first vaccine type that I wanted to talk about is the viral vector vaccines. And just for context, if you are in India, took a vaccine that is called as Covishield vaccine. It is a vaccine that is made by or discovered by Oxford University and commercialized by a company called AstraZeneca. And it uses the very technology, the viral vector vaccines. And I'll just show in a minute how it is constructed and what we are doing in that space. So what are viral vector vaccines and how do they work? So when we think about the virus, a virus is a very, very tiny organism that can do amazing things like killing millions of people. It has tremendous ability to cause disease or death and devastation. But if you think about the biology or the physiology of the virus could also be conceptually understood as a nano vehicle. So it is a very, very tiny little vehicle that can carry genes. That's literally what the virus is doing. So virus is carrying genes that gets expressed in the cells that end up making its own babies. But you could also engineer the virus in such a way that the virus can then be made to carry the genes of your interest that could express antigenic proteins in the body and that could provide the immune response. And that this method exactly leverages the ability of viruses as nano vehicles. So what you do is you identify a pathogen, And in that pathway, so for example, you want to make vaccine to disease X. And then you need to computationally or in experiments identify what is the antigen of choice. What should I use? The most immunodominant or antigenically relevant peptide or protein. Then we take the gene that codes for that particular protein, and then that can then be recombined in a viral vector, which is basically a vehicle that has been engineered to be harmless. So you can basically take out the genes that cause the disease in that particular virus. So at that point, it just became a simple vehicle that is carrying our gene of interest. And when you administer this vaccine to the recipient, what happens is this nano vehicle is taking the gene of your interest into the cells where the protein gets expressed. And the protein is processed in a way that your immune system is able to recognize and produce antibodies. And that is very simply the concept of how viral vector vaccines work. And there are several major viral vectors have been used as vaccine platforms. So there's a whole list of them: retrovirus, lentivirus, and vaccinia virus. And one of the most interesting things that we also work with is the adenovirus. And this has been extensively used in many human clinical trials. And if you also, for the context, the Covishield vaccine, aka the AstraZeneca Oxford University vaccine, uses an adenovirus that is a harmless common cold virus that has been engineered to safely carry the DNA of the spike protein of SARS coronavirus 2. But each of them have some disadvantages with regards to pre-existing immunity. If you use a human adenovirus, for instance, same thing is with the vaccinia virus. But all of these can be overcome, and there are technical advances that we made to address most of these disadvantages. And this is the project that my lab is currently working on to develop a novel adenovirus vector-based vaccine for the COVID-19. And this project is funded by the National Institutes of Health in the US, and in collaboration with our colleagues in Purdue University and US Centers for Disease Prevention and Control, CDC. We are currently working on this project. You can then ask me, since there are already vaccines and which are being used, and why are you now doing research on developing yet another vaccine? So what is the need? So the point is that it is incredible that we have now vaccines in the pandemic to basically address this pandemic. But we also have to keep in mind that this particular virus is not going to go away anytime sooner. Once we end the pandemic stage, there will be an endemic stage where the virus will continue to circulate across the world. And while the current vaccines are helpful in protecting people and to potentially end the pandemic, but we also have to keep in mind that this is an RNA virus that is continued to mutate with a lot of novel variants emerging. And we still need to optimize the vaccines so that they give the maximum benefit of immune response in all groups of people, especially older adults that have weakened immune systems. So, and also to address the pre-existing immunity against the vector in most human beings. So what we are exploring now is to use animal adenoviruses. And again, for context, the Covishield vaccine also uses an animal adenovirus. It uses a chimpanzee adenovirus, but we have other platforms we are using. So we genetically modify the animal adenovirus. The choice is because people do not have any pre-existing immunity against the vaccine vector. And then once we render it unable to cause disease, so now it is a nano vehicle at this stage. And then we engineer the DNA from the spike protein into the adenovirus vector, which becomes a vaccine. But in addition, what we are also doing is we have identified a novel immunogenic peptide from the bacterium that causes tuberculosis. So we will also insert the DNA sequence that codes for this novel immunogenic peptide that we identified, so that the vaccine is now much more optimized to give a robust immune response. And once we create this, this has already been done, and currently we have finished the first preclinical trial in animal models. So the vaccine is then injected so that the modified viral vector vaccine will deliver the spike protein DNA as well as the immunogenic peptide that gets expressed in the cells, and then they instruct our bodies to produce antibodies and the T cell immunity. So what are the major advantages and limitations of the viral vector vaccines? It is a very well-established technology. There is a lot of scientific basis, probably over a decade worth of scientific research backing up these. And one of the incredible advantages is that these vaccines give a very, very strong immune response. And if you heard about the other viral vector-based vaccines produced by a company called Johnson & Johnson, and the J&J vaccine is a one-dose vaccine. So the reason that you don't even need a second dose is because one dose is sufficient to create really robust immune response that can be protective. And even now, you probably heard that the recommendation for the Covishield second dose has been modified. So now you don't have to take a second booster dose immediately in 4 weeks, but you could wait for 12 to 16 weeks. It also comes from the fact that the viral vector vaccines are really strong and robust in using a very, very strong immune response. And more importantly, since the protein is expressed in the cells, they are able to respond, or they're able to trigger the response of both B and T lymphocytes. And again, from your immunology class, you probably remember that the B cells are important for antibody production, and T cells are important for the cell-mediated immune response. And to fight a virus effectively, you need both of those arms simultaneously activated. Then there are, of course, limitations. And one thing that I highlighted in the past is that a previous exposure to the vector, for example, if you use a human common cold adenovirus, then a lot of people in the population may have pre-existing immunity against the vector, and that could reduce the effectiveness. But there are ways in which we can address this. For example, use of an animal adenovirus that most people would not have pre-existing immunity, or to rotate the type of vaccine platform to be used. For example, the Sputnik V Russia vaccine uses slightly a different strategy between the first and the second dose. And the other kind of disadvantage is that they're relatively complex to manufacture. They involve a lot of technical aspects to go into construct the vector and then to manufacture them. So it's not that straightforward or simple. But again, these things can still be addressed as the technologies continue to develop and we have more newer processes being optimized. I will briefly stop here before I talk about the DNA vaccines and quickly check on the chat to see what questions you have. There is a fairly long question from Sunil Kulkarni. It says, how the immune system will express if two different vaccines are given as Covishield 1 first and Covaxin second dose, or first convenience and second Sputnik. So I get the point. We need to understand that the concept of 2 doses is to create what we call as the prime and the boost. And the reason is that the first vaccine will inform or educate your immune system to respond to the antigen that you provided. And the second dose, which is the boost, is going to recall the memory created by the first vaccine. So you have much more enhanced, but long-living and higher antibody response. So basic immunology 101 that you learn, the primary and the secondary immune response. And now the question is, can we rotate, take the vaccine type 1 as a first dose and a different vaccine because the first vaccine wasn't available? So the challenge is that we need to keep in mind that all vaccines are not created equal. And a vaccination is a process of an optimized way to induce your immune response. So these different companies have come up with the dose or the technology or the type of vaccine and carefully optimized in such a way that if you take the first vaccine and boost with the same vaccine, then you get the optimized optimal response. In theory, since most of them also have the spike protein expressing, so theoretically, it may be possible that you could take a company A vaccine first, followed by a company B vaccine. But the challenge is there is no guarantee that they will work as well because simply we don't have the data. So for this to happen, before people start mixing and matching, I think that needs to be studied in the clinical trial so that what combinations would work and what combinations would not work. So in theory, they may work, but I think in practice, that should not be done until there is a scientific evidence that will back the fact that you could mix and match the vaccines. What are the questions we have here? Let me see. What is the scope of developing edible vaccines for animals? What animal species would be most suitable to work upon? And this is a very, not a really novel idea. People have toyed with this idea, but let me tell you, not going into too much details, that this hasn't really worked very well. And some edible vaccines for rabies, for example, in bats that tried through edible baits in the wild, hasn't been very successful. And this could be a completely different topic that I can speak for an hour. But in the interest of time, it is a theoretical possibility, but there hasn't been a lot of advancements for a number of reasons. This has not been progressed as well as we would like. And the other question is slightly on the immunology side, probably a little bit outside the scope, but certainly whether antibody-dependent enhancement of infection is observed with COVID-19 infection in humans, or it's only a phenomenon with coronavirus infection in cats. Why is it so? So ADE is a concept that is known with mostly coronaviruses, and there has been concerns about antibody-dependent enhancements of pathogenicity with the COVID-19. And studies are still being done. If you had previously been exposed to an endemic common cold coronavirus, then COVID-19 infection could exacerbate the infection. And they're still being investigated. We don't have a solid evidence to believe that this is actually happening. But we also don't have solid evidence to rule out that this is not happening. So it's still a work in progress, I would say. How the immune system will express if two different vaccines are given us, I think we addressed that. An antigen can't always be a single, or is it a single pathogen? Maybe there are many virulent antigens capable of quick mutation, so it is most virulent antigen. It's a very long question. So Vikas has a question which is, I think, very complex. If I really understand this, if we take more than one antigen and study and try to produce a vaccine against it, essentially what maybe you're getting at is a multivalent vaccine. Yes, I think it depends on the pathogen. As I was telling earlier, there is no one size fits all. I mean, since coronavirus, you just can take spike protein because we know that is the most relevant protein and then create a vaccine. But for other pathogens, for example, you may need more than one protein. So there is always that choice to be made depending on what makes sense or what is relevant for the pathogen in question. Can we make a vaccine for bird flu as it is transmitting to humans also? It was reported in China. And the answer is yes, there is already a vaccine, or in fact, vaccines. The question is whether or not we in India would want to vaccinate against bird flu. It's a completely different regulatory topic, because vaccines against avian influenza, aka bird flu, have been implemented in countries like Vietnam, for example, Cambodia, with varying degrees of success. A country would decide not to vaccinate until such time that they are able to control the infection by the culling of birds whenever an outbreak happens. Same policy is currently being used in India as well as in the US. Because once you start vaccinating, the country becomes endemic and it has a huge trade implication. So it's a totally different discussion. Does the gene of protein antigen incorporated in the vector always expressed? Are there any chance they are not? A great question, Karthik. The idea is that is the art of designing the vaccine, which I mentioned that they are very complex and difficult in designing. So if the vaccine is properly designed and optimized, and the hope is that the viral vector expresses the protein, right? Otherwise, the vaccine would fail. And the fact that these vaccines are in actual human use tells you that they have all been tested and tried and optimized in a way that they are able to express the protein of interest. Will the vaccines introduced in India against COVID-19 be as effective as the vaccine in other countries? Again, this is a very political question, Shabir, and I don't want to really comment on the differences, but one thing I can tell you is that all these vaccines, despite their differences, they're excellent vaccines to give protection. So it's almost like, you know, you have two different things, but the best vaccine for you or for me is what vaccine I have access to. So at this stage, I don't think there are any major differences in the efficacy. There might be subtle, that is for the scientists to figure out. For common people and for most of us to use in the face of the pandemic, the best vaccine is what you have access to. Which vaccine is the best? I think I answered the question already. On what basis do you choose a viral vector for the generation of the vaccine? This is an interesting question, Akshata. So there are a number of factors will go into it, and some of these viral vectors have already been studied. And one of the choices is about the manufacturability, or how you can engineer them, and how safely we can mutate them to carry. And not all viruses are amenable to manipulation. For example, you can't use some of the RNA viruses like influenza or corona as vectors because they're very difficult to engineer versus adenovirus, for example, or vaccinia virus. So the DNA viruses tend to be more suitable relatively because of the fact that they carry DNA, and you want to carry the DNA that gets expressed. And also several of these platforms have been tested, and more can be developed. Is there an evidence of chromosomal integration of adenoviruses? And no, that is just a concern and a conspiracy that people have portrayed. There is no scientific evidence so far to base that there is any problem in that space. So the last question is about cytokine storm being heard nowadays. Can I get information about? So it's completely out of the context, but what cytokine storm is nothing new, and it is not something that is created or portrayed for COVID-19. We know with a lot of severe viral infections, including highly pathogenic influenza, what eventually causes severe disease and death is how your body is reacting to the virus. And it is the paraphrase is cytokine storm, which is essentially the dysregulated hypercytokinemia, meaning your body is trying to protect you by creating or secreting these cytokines, but they become excessive, and then to the extent that they become harmful. So that is in other words is the immune damage that can be caused to your body. So I'll stop here in the interest of time and move on. But I also wanted to recognize my colleague, Dr. Rma Prasad, joining here, and it's very nice to see his message. Thank you. Okay, moving on. The next topic is DNA vaccines. So what do you do here? So the DNA vaccines, essentially, you basically create a cassette that can carry the antigen of interest. So you basically insert your gene of interest, the antigen genes, and we want this gene to be expressed. So you basically take a plasmid with a promoter, typically a viral promoter that will help the expression of this gene. And then you also want to stop the expression, and there is a terminator sequence included. So it starts here, ends here. So you make an mRNA of the gene, and it makes a protein. And sometimes you can also engineer these to insert other co-stimulatory molecules like cytokine genes. So you're not only inducing the expression of the antigenic protein, but you also stimulate the immune response simultaneously by adding these co-stimulatory molecules. And how do you then inject or deliver these DNA vaccines? So there are a number of different ways. So you can inject a plasmid DNA, for example, or you could create a bacterial vector that has the plasmid DNA and inject it into the cells. or a viral vector that can carry a DNA, which I just mentioned. So in other words, the viral vector vaccines is a type of DNA vaccine. And there is also other method is you can transfect the DNA into the cells, and those cells can be then injected back into the recipient. So all of these different things have been tried. And many of these things are still in research development, and in the years to come, we will see more advances in this space for sure. So how the DNA vaccination works. So please focus on the left-hand part of the panel. I'm not going to talk about the immunotherapy side of it. So basically, you create this DNA vector or the vaccine that I just mentioned. And when this is injected into the body or transfected at the site, so what happens is that this protein is expressed, and it informs your body. So there are specific antigen-specific B lymphocytes and antigen-specific T lymphocytes being activated. And then this will basically traffic into the lymph nodes and creates this robust immune response. Then you have protective response against the antigen of interest, just to keep it really simple. And similarly, this is a bit more expanded version of it. So what happens is, again, going a bit deeper into the immune response. So you want both the antigen to be processed as an exogenous antigen, which will be processed through MHC class 2 molecules. So it produces antibody response. But you also want the protein to be processed as an endogenous antigen, which then processed through the major histocompatibility complex 1, and that will trigger the other arm of the immune response. So the idea is that they will trigger both antibody response as well as the cell-mediated immune response. So you have a much more comprehensive immune protection against the invading pathogen of interest. Again, another cartoon to basically illustrate the same point. The idea is that you want the CD8 positive, the cytotoxic T cell response, as well as the CD4 cells that can then activate the B cells to produce antibodies. And these vaccines are designed to do both of those. And there are a lot of advantages of DNA vaccines, and this is a fairly long list, and I can continue to add to that. The main thing is that they provide a lot of flexibility in designing it. So you could optimize by inserting. So an adenoviral vector vaccine, then you can just take out the spike protein gene from this vaccine and insert a different one, or you could modify or update based on the type of variants that might be circulating. And generally, they are fairly rapid in production and formulation. If you recall my first few slides, I talked about the conventional or the traditional vaccinology. A major limitation is they're very laborious, they take a long time, and they produce a very moderate or small amount of immunogens. But in contrast, the use of DNA vaccines provides this incredible advantage of large-scale production and the manufacturability, which is critical, especially in the face of a pandemic, or if you are trying to vaccinate a large number of animals, for example. And they are very safe because they are completely harmless, and there is no possibility of reverting to virulence. And in contrast to the attenuated vaccines I mentioned, if you modify a live virus in order to lose its pathogenicity but retain the immunogenicity, but those pathogens can always revert back to virulence. But with DNA vaccines, that risk is completely taken away. There is no way they're going to become pathogenic. So the safety profile is pretty high. And they're fairly stable. And they are very temperature stable than conventional vaccines. Especially this is important if you think about the mRNA vaccines like the Pfizer and the Moderna. They require ultra low freezing, but in contrast, the DNA-based vaccines like the Covishield, for example, they're more temperature stable than the other types, and they have a very longer shelf life. And as I said, they induce both the T and B lymphocytes. And there are certainly some limitations and challenges. These are not perfect. And we are still working on those, and many people across the world to make these things better. And there are potential resolutions for many of the concerns. For example, like somebody asked earlier, what are the chances of this DNA vaccines integrate into the cellular DNA and cause any adverse long-term effects. And but the resolution is that the U.S. Food and Drug Administration, FDA in the U.S., it very, very rigorously regulates all vaccines that are meant, any medical interventions for that matter. And they required the manufacturers or the companies to do integration studies for any new DNA products using accepted assays in animals before beginning in human trials. So far, the vaccines that did come into the market have gone through these integration studies to show that they do not integrate in animal models before they're even allowed to do human clinical trials. But this continues to be a concern, and more research is being done to address what are the possibilities and how can they be mitigated. And there are some concerns about the fact that can they develop autoimmune or induce autoimmune disorder against the patient DNA. And again, based on the studies, it is found to be very unlikely. And then there are also concerns about the development of autoantibodies. So the studies also are looking for signs of autoimmunity, and so far there are no evidence to suggest that this could be a major concern. And then the first generation DNA plasmids have relatively poor immunogenicity, but that now has been addressed with the novel formulations that are really very efficient, and especially to the question that somebody asked earlier about the 2 doses. And that is one of the reasons that we do to address that low immunogenicity problem. And you give 2 doses, the prime boost approach, so that the first dose will prime your immune system, but it is still not very strong. But when you boost, the second dose will recall the memory created by the first dose. Now you have a much more stronger and robust immune response that has been studied very well in the clinical trials. And again, in the interest of this group being very heterogeneous, I won't be going into a lot of these details, but the point I'm trying to make is there are a lot of things we could do, or we are doing, to optimize how we can enhance the immunogenicity of these DNA vaccines. So you could optimize the design of the DNA such that the expression is much more efficient. For example, you can select for high GC content of your genome, and then avoid certain things like the Tata boxes and so on. And then you can also formulate adjuvants that can enhance or potentiate the immune response. And then the immune plasmid adjuvants, somebody I think also mentioned in the chat, is that they are the co-stimulatory molecules. For example, the genes that can express for cytokines or the toll-like receptors that also activate your immune response, in addition to the actual antigen itself. Hope I answered that question. So I'll pause again here very briefly to see if any questions on the DNA vaccines before we move on. Okay, there's one question. What is the immune plasmid adjuvant, which I answered? Can we administer DNA vaccines into human cells along with nanoparticles? How far research has been done in this area? Yeah. So the reason we use nanoparticles is you want a vehicle to take the nucleic acid, right? So, and that is a very good question to follow on to the next topic I'm going to talk about. Why do we need nanoparticle vehicle for the mRNA vaccines? Because DNA vaccines are relatively stable, because DNA, as we all know from our basic molecular biology, DNA is fairly stable in the lab, but RNA is very, very fragile. So if you really want to use RNA as a vaccine, then we also have to make sure that it is stable and it can be delivered into the cells without, basically destroying the RNA. So we'll see what can be done with the RNA. But for DNA vaccines, we have ways to deliver the DNA, for example, plasmids or viral vectors. So we don't necessarily need the nanoparticles. But certainly, this is also an area that is being tried to package the DNA in a nanoparticle. That is theoretically a possibility if we need to do so. So the next topic is the messenger RNA vaccines, which is, I think, the most technological advancements that we saw in the past year and a half. Just to emphasize how incredible this is in the science is that we have never heard in the history of medical vaccinology that a vaccine has been designed and developed and administered to millions of people in just 1 year. And thanks to this research that enabled this development, and then the messenger RNA vaccines, the two companies that have produced these are Pfizer, BioNTech, and the Mrna. The point is that now that these vaccines have been tested and used in people and got the emergency use authorization, now the field is rapidly moving towards application of mRNA-based therapeutics, not just vaccines, but a whole bunch of applications in human medicine. So in the next 5 years, perhaps, the mRNA interventions in the medical countermeasures is going to be significantly the leading player in the field. So what are mRNA vaccines? mRNA vaccines are designed to instruct the body's cells to make an antigenic protein. So think about your basic cell biology. So there is DNA that has the genetic sequence, and the sequence on DNA is transcribed into a messenger RNA. So that is the transcription. And the messenger RNA then translates into protein. So you have transcription and translation. So the idea here is that if you use the DNA vaccines, you're starting from the first step. So the DNA goes into your cells. You need a promoter to transcribe the DNA into messenger RNA, and then the messenger RNA gets translated into proteins. But if we can cut down the first step and basically start from the second step, meaning you already start with the messenger RNA, so you don't need the transcription, so that the messenger RNA directly translates into proteins. And that is the fundamental concept of messenger RNA vaccines. So how do you design a messenger RNA vaccine? So we need a messenger RNA expression plasmid that's called pDNA that encodes a DNA-dependent RNA polymerase promoter. And these are typically derived from bacteriophages. And the RNA vaccine candidate, we design a template for in vitro transcription. So basically, you take the DNA of spike protein, and then in the lab, in vitro is in the lab, you transcribe the RNA from the DNA. So now you have the RNA that encodes spike protein, for example, in this case. And the antigenic sequence is flanked by a 5' and 3' UTRs, the untranslated regions. And then you can insert a poly A tail from the 3' end to the template, or you could add it enzymatically after in vitro transcription later on. So these are some of the fine details of how you design them. But the concept is simply you need to have an in vitro transcript RNA that is flanked by the UTRs, and then we can then deliver it to the cells. Then how do we deliver is the next question. And there are two types of RNAs that are currently studied as vaccines, and they fall into these two buckets. First one is non-replicating mRNA. So the mRNA is what you give is what you get. Whereas you could also use the virus-derived bits and pieces to make a self-amplifying RNA. So the RNA is amplified so that you have a much more antigen availability in the cells. So the conventional mRNA vaccines simply are non-replicating, and the current COVID-19 vaccines are, just to clarify, are non-replicating mRNA vaccines. But the research on the self-amplifying RNAs is still continuing. And the difference is that the self-amplifying RNAs encode not just the antigen, but also the viral replication machinery that enables intercellular RNA amplification and abundant protein expression. I just wanted to check, are we doing okay for the time?
Yes, sir, it's fine. You can continue.
Okay, thank you. So again, not going into too much details, but the idea is that you take the vaccine immunogen and flanked by a 5' and 3' untranslated regions and add poly A tail. And when you give this to the recipient, there will be in situ translation. So the recipient, so if I take the mRNA vaccine, the mRNA will then translate into protein. So in this case, say if we talk about the COVID-19 vaccines, it leads into spike protein expression, and then that will create the immune response if it is vaccine, or you could also use it for immunotherapy. And the mRNA 1273 vaccine candidate is produced by a company called Moderna. I just wanted to give an example of how this was done. And to one of the questions that people asked earlier, the RNA cannot be just given naked because they are extremely unstable and they'll be destroyed by the cells. So you need to optimize the delivery and the length of expression in the cells. So they are encased in lipid nanoparticles. In this case, they're composed of 4 different lipids. And the mRNA lipid ratio is also optimized so that you will get enough antigen supply in the cells, and then enough, or the duration can be regulated. So you could also design the lipid nanoparticle ratio with the mRNA in such a way that you can optimize how much protein you want to be expressed in the cells and how long you want the mRNA to be active. All of those can be optimized. And the other concept is about the self-amplifying RNA, which is a step up from the non-replicating RNAs, if you will, is that it is still the same thing. You have the vaccine immunogen, but that is flanked by an alpha virus non-structural protein. And then you also have a CSC, which is a conserved sequence element that will help in the creation of the RNA-dependent RNA polymer in the cell. So basically, you're creating a situation where this mRNA is amplified amplified in before it is injected. So you're basically creating a system to amplify the RNA, and then this can be injected for translation. So the difference is that in the non-replicating, you just have one strand of messenger RNA that is not amplified, but here you have the viral enzymes inserted, so you are also amplifying the RNA to get enough antigen. And similarly, there is another concept of trans-amplifying mRNAs, which is very similar to what the conventional mRNA and then what the self-amplifying RNAs do. But the difference is that you basically, rather than one single piece, you create these in 2 separate pieces. So you have the alpha virus NSP in a separate segment, and then you have the vaccine immunogen with the CSC sequence, and then the in situ translation will create this RDRP, and the end result is very similar. You amplify the RNA, and then there's protein. So again, these are all advances in development, and there will be a lag time before these could become tools that are used in the medical practice, and probably much later in the veterinary practice. So now, what are the benefits of mRNA vaccines? I did emphasize some of them. For example, the production. So mRNA vaccines have the potential for very rapid, inexpensive, and scalable manufacturing due to the high yields of in vitro transcription. You don't have to grow a virus. You don't have to grow the bacteria, but you can create this RNA in the lab in very, very high quantities. And that has really helped in the massive manufacturing of vaccines, especially the mRNA vaccines that are used in the US. And these vaccines are very safe because they're non-infectious, non-integrating platforms. So, and one concern with the DNA is the DNA may then integrate into the host chromosome. With RNA, that possibility is also not there. So that concern is eliminated. And then their mRNAs are easily eliminated or degraded by the normal cellular processes. So they're only there for a very, very brief transient period that we optimize. And after that, they're completely gone. The cell will just take care of them. So, and then the beauty is that you can really regulate the half-life of how long you want them to be expressing in the cells. And that gives us an extremely good leverage to optimize and so that they can be efficient at the same time and also safe, so you don't have an RNA lingering in your body for a long period of time. And as we know now, by vaccinating millions of people with these vaccines, they're very, very efficient in creating this strong antibody response. At least the companies have claimed, based on their data, that these vaccines are the efficacy rate is as high as 90 to 95%. And one of the things that I mentioned about the viral vector vaccines, for example, the Covishield or the Johnson & Johnson vaccine, is that there is this concept of the immune system also responding to the vector. So you are trying to use the viral vector as a vehicle, and your intention is to get the viral antigen expressed, but your body is also seeing the viral vector and it is also responding to the viral vector. So, and subsequently, when you want to reuse the same vector, there will be some previous anti-vector immunity that might undermine the efficiency of the vaccines. And there are, again, other things are being done to overcome that. But with the mRNA vaccines, you don't have that problem, because all you're doing is you're taking a messenger RNA, package in lipid nanoparticle, just give it. So there is no vector involved, so there is no anti-vector immunity. So that the problem solved. And so that mRNA vaccines can be administered repeatedly. So you could give any number of booster doses without having to worry about the second or third dose being not as effective as the first dose. And again, one limitation currently is that because of the nature of their stability, they require ultra low temperatures like -70 or -20, which raises some logistical challenges to implement these vaccines in many countries where we don't have the type of resources or cold chain like in the Western world. But again, research is being done to make sure that those problems can also be addressed. And this is a bit more scientific that I'm not going into, but the point is that there are a lot of considerations that are being done in research to optimize. It's not simply you create an mRNA and insert it into the cells. A lot of scientific rigor goes into how you optimize them. So you can optimize the vaccine to choose how efficient your carrier is, and then you can create the immune stimulatory approach so that there is a better immune response and what kind of antigen presenting cells you're targeting. And at every stage, you can design the vaccines to get these optimal responses so that the vaccine is very, very efficient and induces a very strong protective immune response. And now switching gears to where are we in the veterinary vaccines field? Before I go into this, maybe quickly check on to see if any questions. What are the different diseases in which the RNA-based therapy may have potential application? Excellent question. And there can be, now all the companies are opened up to this new reality of using mRNA in the therapy, and there is now incredible speed in this. And one quick application that is currently being tried and probably will come into market very soon is the cancer immunotherapy. And there are many, many other immunotherapies that are being investigated. So not just the vaccines, but the mRNA technology is potentially going to revolutionize the human medicine as we know. Next question is, which vaccines are most likely used? Vector vaccines or mRNA vaccines? Not quite sure if I understood the question, but I think if the point is that when do you choose a vector vaccine or an mRNA vaccine, it again depends on the considerations you have to make about how do you design a vaccine. What is your technical capability or who is making these vaccines? So all of those considerations, and then the facilities for manufacturing and scalability, that would essentially inform what platform you want to choose for developing a vaccine. So that is a completely different set of criteria you want to discuss to decide which is the best vaccine platform in a particular setting. Does mRNA vaccines lead to long-term effects like Alzheimer's disease? And as I mentioned to you, the answer is no, based on all we know about how mRNA vaccines or mRNA therapeutics work, because they're only there for a very, very short period of time, and your body or your cells will degrade the RNA. So it is highly unlikely that they have any long-term effects because it's just not there. It's gone very, very quickly. Okay, now getting back to the presentation. So this is as of 2017, 2018, all the different advanced or recombinant veterinary vaccines. And one thing you might quickly notice is that there are only 2 DNA vaccines of all the different vaccines. And predominantly they are either viral vector vaccines or subunit vaccines, and only 2 DNA vaccines, and one in horses and one in fish against infectious hematopoietic IHNV virus that we also work here with. So the point is that there is so much potential to accomplish in the veterinary, and we always tend to be a step behind the human medicine for a number of practical and legitimate reasons. But I think one thing that we are very hopeful that the COVID-19 pandemic has really accelerated the technological developments in diagnostics and vaccines. So very soon, a lot of these technologies will percolate into the veterinary practice, and we can hope to see a lot of nucleic acid based vaccines in the veterinary practice. But currently, there is a large limitation in how many different recombinant type of vaccines we're currently using. And there are also legitimate food safety concerns, and people are, if you give a modified recombinant DNA type vaccine, what will happen to the meat or the milk? So there are some food safety concerns that need to be addressed, unlike in human medicine. So, but all of those, I think, will be resolved, because the key driver here is the technological advancement is really opening up the possibilities that we were never aware of. And it is very difficult to stop these technologies to transform the medicine as we know. So very soon, if not immediately, we'll start seeing these technologies to also transform the veterinary vaccinology. And we'll probably, if I have to give this talk in 5 years' time, perhaps I may have a slide with a lot of DNA or mRNA vaccines in the veterinary application, and let us hope so. And finally, why is this? We always talk about these technologies and we learn, and it is very fascinating to understand, but they are not really practically implemented as well as they are being implemented in the human medicine. Because the fact that the first slide, if you recall, I showed you that the veterinary medicine as a field is very, very complex, because we are not dealing with one patient, but we have a whole range of different patients that are completely different. So naturally, developing a vaccine for veterinary application is inherently challenging because of multiple species and a whole bunch of pathogens. And the most important driver, perhaps in the veterinary field that is different in the human field, is the cost of the vaccine. So you can't spend 500 rupees per a vaccine to a chicken, but you could pay 500 rupees to get a vaccine for yourself. So the larger market barriers in the veterinary field is the cost. So you have to make sure that the cost of the vaccine dose is amenable or alignment with the cost of production of the production animals. And that is also a key driver in the technological development of how can you make these technologies more affordable and cheaper, so that we can also use them for vaccines in veterinary applications. And the other important thing I wanted to really emphasize to this particular group, because this is a fundamental obligation for all of us as veterinarians and scientists, is to recognize that there is this vaccine hesitancy. And unfortunately, the scientific information doesn't get spread as quickly or as easily as misinformation spreads. So the fact that we have this incredibly effective vaccines against COVID-19 in unprecedented time of less than a year hasn't really excited people. But then the fact that there are 5 or 6 or 7 people that had developed clots after taking a vaccine, and that was given to several millions of people, had really spread like wildfire. So it is really important to recognize the risk analysis or risk benefit ratio is what are the odds of dying from COVID-19 versus what are the odds of getting blood clots, for example. And similarly, I'm sure you all have experienced that there is also a big vaccine hesitancy among livestock producers across the world. Some people believe that vaccination would reduce the production of their cattle, for example. So And I think the important thing is to recognize that there is vaccine hesitancy both in the human and the veterinary medicine or the application. And we as veterinarians and educators and scientists have the obligation to educate people in our own circles so that they understand the science and not swayed by this misinformation. And this is now very critical than ever before because of the explosion of social media. And now people believe if something comes in WhatsApp, it got to be true, right? If somebody says you eat banana and you can cure COVID-19. And we have no idea who creates this, but people believe that if it is in WhatsApp, it got to be true, which is not the case, as we all know. So the importance of education cannot be emphasized enough. And I really wanted to highlight to this group that please think about vaccine hesitancy, and in your capacity as a faculty or a veterinarian or whatever the role might be, try your best to help mitigate this vaccine hesitancy to the extent possible. And as I also mentioned that we believe that the COVID-19, the positive side is it opened up our imaginations to incredible opportunities that we haven't been thinking in the past. So these opportunities, I believe, will also drive the advances in veterinary vaccinology. And especially the DNA and synthetic RNA vaccines provide a novel solution to develop vaccines against emerging pathogens. And always keep in mind that we cannot use the good old 18th, 19th century technology to make vaccines against a raging pandemic or panzootic, if you talk about an animal disease, because we need a large supply of vaccine in a very, very short span of time. And to be able to distribute to the scale that is possible, if you still rely on having to grow the virus or grow the bacteria, inactivate it and create a vaccine, you're simply not up to the game because you're no longer going to produce enough doses. So that's why it is absolutely critical that rather than being cynical about, oh, this DNA is going to integrate into your body or it causes Alzheimer's, all these concerns, but look at the bright side of it. This innovation really helped us to create these pandemic vaccines at a dose and the scale that is required to vaccinate billions of people across the world. If we simply rely on our conventional, traditional vaccine approach, there is no way we're going to make enough vaccines in any reasonable length of time. And the vaccine is no good if it is only available after 2 years after the pandemic, because most of the devastation is already done and it is really not useful. So you need to put off the flames as the fires are raging. And the only way we can do this is by innovation and embracing innovation. And part of that is also on all of us to try to implement these technologies in our own work or wherever feasible and try to do research in this area, but more importantly, educate people that the innovation is really helpful and it is not something that is going to damage our health. If anything, it actually makes our lives better. And with that, I would also want to recognize a whole bunch of people that work in my lab, and whatever I have told you or said to you is because of this incredible people that work in my lab manager and the postdocs in the lab and my PhD students and technicians. And I also wanted to identify and acknowledge a whole bunch of my collaborators from many, many universities across the country, especially the Houston Methodist Hospital, Purdue University, CDC, and the Army Research Laboratory. And I'm part of the Penn State Huck Institute of Life Sciences. And as I mentioned, my research is funded by the 3 federal agencies in the US, NIH, NSF, and USDA. With that, the final thought is I wanted you to take away today is that vaccines save lives. If you have not taken so, please get a vaccine if you have access to one. And please also encourage people to get vaccines if they can access one. And the vaccination of all people or most people is the only way we can end this pandemic. With that, I once again want to thank you for participating, and I'm very happy to answer any questions if you have.
Thank you, Dr. Suresh, for a very elaborative lecture on the vaccinology what are different avenues of the vaccines, different types of vaccines which can be there, and its correlation. Perfect examples you have given every time, correlating with the current COVID pandemic and all. Excellent one. Now, I would request my co-chairman, Dr. Deshmukh, if he could find some questions which are not answered from the chat box, please pick up and ask to the speaker. Dr. Deshmukh, are you there?
Yeah. Now, most of the questions Dr. Suresh has already answered from chat box himself, because he was interactive all the while during the session. And on behalf of our university, I thank him. He has given elaborately all the pros and cons, new technologies available. Only last point he made, there is a contribution of veterinarians in development of vaccines, 25 days in Moderna, Pfizer development, designing of mRNA, etc. All over the world, veterinarians, they are contributing. For the knowledge, I would like to say here in India also, we are using traditional platform of inactivated vaccine. And what is the role of veterinarian? So latest is that we have augmented our production capacity, and recently all public sector vaccine production units like Indian Immunologicals, PIPCOL, and private one like Intervet facility at Pune or in BioVet in Bangalore, they are using old fermentation technology for production of inactivated vaccines. And this way, there is a one section in Indian society who is cost conscious. And for that cost conscious and from social point of view, I think that proven technology also needs to be given chance in this pandemic. And as far as this today's context is concerned, the RNA and the DNA type of the vaccines, these are called as new generation vaccines. And you have given, because the group is heterogeneous, and for their understanding, I would appreciate your presentations, particularly the figures.
It's like the audio got cut off. I can't hear anything.
I think he lost the connectivity. Anyway, he was appreciating your lecture, and I do agree, and I endorse the thing that today both lectures were par excellence, and they made the very you both made the very complex issue simple, understandable. That's why we could have so many questions on it. Many of the things probably just we read it now in the social media or in some articles, but you made it, both of you, with your diagrams and all things were made very simple and very elaboratively. Everyone took it very nicely. That's why these questions and interactions was more. So from
Can I just
chairman
make one
and
more
co-chairman.
comment?
Yeah, please.
Sorry, if you don't mind. So just a couple of comments. So to the point that Dr. Deshmukh made, I think this is a very, very important aspect that we all need to recognize, that let us not the perfect be the enemy of the good. So the idea of new technologies and innovation is that they are not necessarily mutually exclusive. So it never works that they completely replace what we have historically or conventionally done. But the idea is that they're going to complement, and if anything, make the process much better. And just example that from Dr. Jayaral's talk this morning, if you thought about veterinary diagnostics in probably 10, 15 years ago, the thought of PCR was very, very new to people, and they were very skeptical that, oh, I still need to isolate my bacteria, and how can you tell a small piece of nucleic acid if it's present or not? But if you now look fast forward 10 years, now PCR is became the bread and butter of veterinary diagnostics. This is literally what we do. So I think there is always a transition period from the conventional vaccines or conventional technologies that have been tested and useful, but then gradually they'll be either partially or completely replaced by more efficient methods. So we are in that transition period where we need to recognize the older methods are still useful, but then we also have to open up our imagination for the new innovation. And I also wanted to thank you all and the organizers for this incredible conference program to put this together. And I feel that all the participants may have really benefited from this outstanding lineup of topics that you all have assembled. So thank you for organizing this. It's been an incredible opportunity for me to talk to this group and very, very interactive audience as well. Thank you.
Thank you. Now over to the organizers. From my side, it's over now. It's already been 30 minutes or 10, 15 minutes over the time. You both may be guys in hurry to rush to your working place. It's morning now. I don't know the time, but must be 10 o'clock or something like that. So we took much time. Due to interaction, the time was taken more. So now over to the organizers for vote of thanks. Dr. Jende, please.
Yeah. Thank you, sir. Thank you so much. I first of all would like to thank Dr. Jera Bhushan sir and Dr. Suresh for a very informative and very elaborative talk on both the topics what they have an expertise. And definitely our participants will benefited out of this both the lectures. And really I made our honorable director of research, particularly as I know he is very scientific and he will not spare the timings. And the things are that you know that just going on very good informative lectures you both have delivered. And thank you so much also for our director of research, chairman and co-chairman Dr. Vivek Deshmukh sir, for conducting smoothly in the today's session and interacting with all participants in both the speakers. So thank you so much, sir, once again. And thank you, sir, Bhushan sir, and Dr. Suresh. Thank you.
Thank you. Yep.
Yeah. Sonal.
All do take care in this unusual pandemic. Thank you. Have a good time.
Okay, sir.
Here. Yep.
Thank you. Bye-bye.
Thank you, sir. And to all the participants, I would just like to mention tomorrow again we will meet at 11 o'clock in the morning. So tomorrow's session we'll be having in the morning. With that, we will conclude today's session here. Thank you all both the speakers and the dignitaries for chairing the sessions, and all the participants to please for their patient hearing. Thank you, sir.
Thank you. Thank you. Thank you so much.